One woman's cancer cells have been alive since 1951. Another cell line helps produce vaccines used by millions of people worldwide. And one of the most important cell factories on Earth comes from the ovary of a Chinese hamster. Welcome to episode 7 of the Cell Culture series, the famous cell lines, HeLa, HEK293, and the gang. Today, we're meeting the cell lines that changed biomedical science forever. A woman, a cell line, and a revolution. In 1951, a 31-year-old black woman named Henrietta Lacks walked into Johns Hopkins Hospital with cervical cancer. Her tumor cells were taken without her knowledge or consent. Those cells became HeLa, the first immortalized human cell line, the foundation of the polio vaccine, and still dividing in labs worldwide today. Her family did not find out for decades. Why this matters beyond the lab? The HeLa story fundamentally changed biomedical ethics. Informed consent is now legally mandatory for research using human biological material. Every consent form you will ever sign as a research participant exists partly because of Henrietta Lacks. Science owes her an enormous, largely unacknowledged debt. Before we move on, let's test our understanding with a quick question. What makes HeLa cells immortal compared to normal human cells? Take a moment and think about it. The answer is that normal human cells can only divide around 50 times before they stop growing, a phenomenon known as the Hayflick limit. HeLa cells are different. Because they originated from a cancer, they carry genetic changes that allow them to bypass this limit. Their telomerase enzyme remains active, continuously maintaining chromosome ends, while important growth control pathways such as p53 are disrupted by HPV-related proteins. As a result, HeLa cells can continue dividing indefinitely under the right culture conditions. It's important to remember that this isn't a special ability unique to HeLa cells alone. It's a characteristic shared by many cancer cells, which is exactly why they became one of the most important tools in biomedical research. The All-Stars, 6 cell lines every scientist knows. Let's meet the All-Stars of cell culture, 6 cell lines that have shaped modern biomedical research. First, HeLa cells *, derived from Henrietta Lacks in 1951. They're the world's first immortal human cell line and have contributed to everything from polio vaccine development to cancer and COVID-19 research. Next, HEK293 cells, originally derived from human embryonic kidney cells and modified with adenoviral DNA. They're widely used for recombinant protein production and have played a major role in modern vaccine technologies. Then there's *CHO cells are Chinese hamster ovary cells. If you've ever heard of therapeutic antibodies, insulin, or other biologic drugs, chances are CHO cells helped produce them. Vero cells *, derived from African green monkey kidney, are a cornerstone of vaccine manufacturing and have been used extensively for viruses such as rabies, polio, and influenza. Jurkat cells * are human T-cell leukemia cells and remain one of the most important models for studying immune responses, T-cell signaling, and immunotherapy. And finally, NIH3T3 cells *, mouse fibroblasts that became a classic model for understanding cell growth, oncogenes, and cancer biology. Each of these cell lines has a unique history, but together they form the foundation of countless discoveries in modern life science. One of the most important decisions in cell culture is choosing between * immortalized cells * and * primary cells. Immortalized cell lines can divide indefinitely, making them cost-effective, easy to maintain, and highly reproducible. That's why they're widely used for basic research, high-throughput screening, and biopharmaceutical production. Primary cells, on the other hand, come directly from living tissues. They have a limited lifespan and are more challenging to culture, but they provide a much more realistic representation of what happens inside the human body. In simple terms, if you need consistency, scalability, and convenience, immortalized cells are often the best choice. If you need physiological relevance and clinically meaningful data, primary cells are usually the better model. Neither is universally better. The right choice depends entirely on the scientific question you're trying to answer. Let's test your understanding with another important question. Why are CHO cells the dominant platform for manufacturing therapeutic proteins instead of E. coli? Take a moment to think about it. The correct answer is B. E. coli cannot perform the post-translational glycosylation that many therapeutic proteins require. Here's why that matters. Many biologic drugs, including monoclonal antibodies, erythropoietin, and clotting factors, are not just proteins, They also need specific sugar molecules attached to them through a process called glycosylation. These sugar modifications help proteins fold correctly, remain stable in the bloodstream, and perform their intended biological function. While E. coli is fast, inexpensive, and easy to grow, it lacks the cellular machinery needed to carry out these complex modifications. CHO cells, on the other hand, can produce proteins with glycosylation patterns that are compatible with human biology, making them the preferred choice for biopharmaceutical manufacturing. That's why, despite being more expensive and challenging to culture, CHO cells remain the industry gold standard for producing therapeutic proteins. Read your cells, morphology and the passage number rule. One of the most useful skills in cell culture is learning to recognize cell lines by their appearance under the microscope. HeLa cells typically show a cobblestone-like epithelial pattern, with large irregular cells and prominent nuclei forming a dense monolayer. HEK 293 cells look quite different. They tend to form loose clusters and often show a semi-suspension behavior, creating an uneven monolayer. CHO cells are usually smaller and more uniform, with clean cell boundaries and a well-organized appearance. NIH3T3 cells are classic fibroblasts. They're elongated and spindle-shaped, and as they grow, they align with one another while maintaining strong contact inhibition. Jurkat cells are suspension cells, meaning they never attach to the culture surface. Instead, they appear as round floating cells that often form loose aggregates. And finally, A549 cells display a cuboidal epithelial morphology, appearing larger and flatter with a prominent nucleolus and characteristic lung epithelial packing. With experience, these visual patterns become instantly recognizable and help you quickly assess whether your culture looks healthy and authentic. Remember, before running any experiment, your first quality control tool is always the microscope. Passage number. The silent experimental variable. Every passage applies selection pressure. Cells that grow fastest under your specific conditions outcompete others. Over many passages, the genetic composition of your culture drifts from the original. Passage 15 and passage 80 can produce dramatically different experimental results, but they look identical under the microscope. The passage tracking rule. Write passage number on every flask. Record it in every experiment entry. Check ATCC datasheet for the maximum passage number recommendation. Never exceed it for critical experiments. When approaching the limit, thaw a fresh authenticated vial from your working bank. Now let's test your understanding with a real-world lab scenario. Imagine your HeLa cells still look perfectly healthy under the microscope. The morphology is normal, growth seems fine, and there are no obvious signs of contamination. But over the last 6 months, your drug response data has gradually started to differ from what's reported in the literature. What is the most likely explanation? A. The drug has degraded during storage. B. Genetic drift from excessive passaging. The cells have lost their original phenotype. C. Normal biological variation between experiments. D. The published data was probably wrong. The correct answer is B. Genetic drift from excessive passaging. This is one of the most common causes of irreproducible results in cell culture. The tricky part is that the cells can still look completely normal. However, after many passages, small genetic and epigenetic changes accumulate over time, gradually altering cell behavior, gene expression, and drug sensitivity. That's why experienced researchers maintain strict passage limits and regularly return to low passage cells from a working cell bank. If your results start drifting, the best solution is often to thaw a fresh, authenticated vial and compare the response again. Remember, cells don't have to look different to behave differently. 3 mistakes and the global stakes. Mistake 1. Using cells beyond recommended passage number. The fix. Set a maximum passage number before you start work with any cell line. Check ATCC recommendations. Bank at low passage. When you approach the limit, thaw fresh. Document every passage number in your records. Mistake 2. Using another line's culture conditions. CHO cells in HeLa media. HEK 293 cells at HeLa CO2 conditions. It seems harmless but produces slow growth, abnormal morphology, and unreliable biology. Every ATCC datasheet specifies medium, serum percentage, CO2, and split ratio for a reason. The fix. Download and read the ATCC datasheet for every new cell line before touching it. 2 minutes reading prevents weeks of unexplained results. Mistake 3. Treating bioethics as a box to check. The HeLa story is not ancient history. Cell line misidentification has been found in an estimated 15 to 20% of published biomedical research. Patient data governance, genomic consent, and indigenous research ethics are live in contested fields today. Being a responsible scientist means engaging with all of this. The fix. Read The Immortal Life of Henrietta Lacks by Rebecca Skloot. Understand your institution's human subjects requirements. Know what STR profiling is and why authentication matters. Ethics is part of your technical training. Let's finish with a final challenge, a scenario that has unfortunately happened many times in real research laboratories. Imagine you've been working with what you believe are HeLa cells for the past 18 months. You've generated data, run experiments, and built an entire project around them. Then you perform STR profiling, and the results come back. The cells are not HeLa. Their genetic fingerprint matches MCF7 breast cancer cells instead. So what's the most accurate description of this situation? A. Asterisk no problem. HeLa and MCF7 are essentially the same biology. B. The STR test must be wrong because the cells look like HeLa under the microscope. C. Asterisk classic cell line misidentification. Your experiments were performed on the wrong cell type, and the results must now be re-evaluated in the context of MCF7 biology. D. Asterisk only experiments directly comparing HeLa with other cell lines are affected. The correct answer is asterisk C. This is a textbook example of cell line misidentification, a problem estimated to affect between 15 and 20% of biomedical research. The important point is that the data itself isn't automatically useless. The experiments were real, and the results may still be scientifically valuable. The problem is that they represent asterisk MCF7 biology, not HeLa biology. Asterisk. Because some cell lines can appear surprisingly similar under routine microscopy, visual inspection alone is never enough to confirm identity. That's why STR profiling is considered the gold standard for cell line authentication. The lesson is simple: always verify your cells before building years of research on them. A single authentication test can prevent months, or even years, of misleading conclusions. Episode 7 Summary. What you now know. As we wrap up Episode 7, let's review the 3 key lessons every cell culture scientist should remember. First, immortalized and primary cells serve different purposes. Immortalized cell lines can divide indefinitely, making them ideal for routine research, mechanistic studies, and large-scale screening. Primary cells, while more difficult to maintain, provide a more realistic representation of what happens inside the human body. The best model depends on the question you're trying to answer. Second, every cell line has its own requirements. Culture medium, serum concentration, carbon dioxide levels, and passage limits are not suggestions, they are part of the protocol. Always read the datasheet before working with a new cell line. A few minutes of preparation can prevent weeks of troubleshooting. Third, remember that every cell line has a story behind it. The history of Henrietta Lacks transformed how we think about informed consent, patient rights, and research ethics. Good science is not only technically accurate, it is also ethically responsible. Before moving on, make sure you're comfortable explaining the 6 famous cell lines and their major applications, the differences between immortalized and primary cells, why CHO cells dominate biopharmaceutical manufacturing, why passage number tracking matters, and how the story of Henrietta Lacks continues to influence modern biomedical research. If you can confidently answer those questions, you're ready for the next step in your cell culture journey. Thanks for watching Episode 7 of the Cell Culture series. In the next episode, we'll dive deeper into advanced cell culture techniques and the practical challenges researchers face in real-world laboratories. See you there. If you found this episode useful, don't forget to subscribe our channel. Don't forget to like, share, and leave the comment below. We'll see you in the next episode. Thank you for watching.